Here we will discuss the models of embryology. So I have tried to explain the different models with labeling and the important questions which can be asked during the viva on the models of the embryology. So first model is of the development of villus, showing the primary, secondary and tertiary villus. In this the cytotrophoblastic core surrounded by syncytial trophoblast, so that is the primary villus. Then the mesodermal core is coming in the center, surrounded by single layer of cytotrophoblastic cells covered by syncytial trophoblast, that is secondary villus. Then the mesodermal core is traversed by the villus capillaries, then it is surrounded by this cytotrophoblast layer, then syncytial trophoblast, so that is the tertiary villus. First and second, or primary and secondary villi, they appear during second week, and the tertiary villi appear during the third week. So what are the important questions which can be asked on this model is, what are the different types of chorionic villi? Your answer will be primary, secondary and tertiary. So what is the difference between primary, secondary and tertiary? So primary villus has core of cytotrophoblastic cells surrounded by syncytial trophoblast. Secondary villus has core of mesoderm covered by single layer of cytotrophoblastic cells, which in turn is covered by syncytial trophoblast. Then tertiary villus is same as of secondary villus, means all these structures are present, but only difference is that mesodermal core is traversed by fetal capillaries. Then is what are the functions of chorionic villi? So basically they maximize the surface area that comes in contact with the maternal blood for exchange of gases, nutrients, etc. Now this is the second model showing the placenta in the second half of pregnancy. So in this model, what is shown is that chorionic plate is visible on the fetal side and decidual plate is visible on the maternal side. So this is the maternal side of the model, placenta, and this is the fetal side of the placenta. Now on the maternal side, this decidual plate is sending the septi, which is called as decidual septum, and these septi, they divide this space which is lying between decidual plate and chorionic plate. This space is known as intervillus space, and that is divided by these septi into different compartments. We call them as cotyledons. Now what are the other structure visible over here is, this is the spiral artery. So endometrium, they are lying in the endometrium. Then the chorionic vessels, they converge to form the umbilical vessels. Then endometrial veins are visible. So these structures are visible in this model. Now this placenta is the full term placenta. So which side? That is fetal side of full term placenta, showing the chorionic plate and chorionic vessels. So as we have discussed, this is the chorionic plate on the fetal side, chorionic vessels are visible. So this side is the fetal side and this is the maternal side. So umbilical vessels are visible. So this is the full term placenta. Now if we see what is the shape of placenta, it is discoid shape with a diameter of 15 to 25 centimeter. Now what are the important questions which are coming out from these two models is, that is what are the components of placenta? Your answer will be fetal portion and maternal portion. Maternal and fetal sides are covered by which structure? Fetal side is covered by chorionic plate and maternal side is covered by decidual plate. What is the name of structure present between chorionic and decidual plate? Intervillus space will be the answer. What is present in intervillus space? That is maternal blood. What is the site of attachment of umbilical cord? It is usually eccentric or occasionally marginal. You can see in this model it is eccentrically attached, or it may be attached at the margin. Now what are the functions of placenta? So these are the functions. Then congenital anomalies of placenta, they are depending upon the shape, adherence and site. Shape is, these are the variations, that is bidiscoidal, lobe, diffuse placenta succensurata, fenestrated placenta, circumvallate placenta. Adherence, how it is attached to the wall of the uterus. So placenta accreta, then increta and percreta. So site anomaly, it may be attached near the os, we call it as placenta previa. So these structure, that is succensurata, fenestrated, circumvallate and accreta, increta, percreta, the detail of these structures you can study from your textbooks. I am just telling you what are the important questions which are related to that particular model. Now model 4 is showing the stages of the development of the fetus along with full term placenta. It is showing the sperm, it is showing the section of the ovary, it is showing the primary oocyte, it is showing the secondary oocyte with first polar body, then the fertilization is shown, then breakdown of the membrane of the pronuclei, that is shown. So first and second polar bodies are formed because after fertilization the second meiotic division is completed. Then the cleavage spindle, they are shown over here. Then in this, the development of the fetus in the uterine cavity is shown. So in this model, you can see the pharyngeal arches, they are visible. Then growth of the fetus is happening over here. This is the fallopian tube, this one is the fimbriated end of the fallopian tube, and this is the ovary, you can see. So this is the fallopian tube. Now development of the fetus is shown. This is the wall of the uterus, this is the full term placenta, this is a section of the full term of placenta. So this is which side of the placenta? I have told you, this is the fetal side, showing the chorionic vessels, chorionic plate. Then amnion is present, this white color structure, amnion is there. Now what are the important questions which are coming out through this model is, that is what are the parts of sperm, because section of the sperm is visible. So what are the question can be asked, that what are the different parts of sperm? It has head, neck, middle piece and tail. Head is covered by acrosomic cap. Then what are the stages of spermatogenesis? From spermatogonia, primary spermatocyte is formed, then secondary is formed, then spermatid and spermatozoa. In this you can see the level of first and second meiosis, and spermiogenesis is basically the conversion of spermatid to the spermatozoa. Now you can see here primary spermatocyte has the diploid number of chromosome, but secondary spermatocytes, they have the haploid number of chromosome. So this is the reduction division. Then is at which state of spermatogenesis the chromosome number is halved, as I have told you. So when the primary spermatocyte is converted to secondary spermatocyte, the number of the chromosome, it is halved. How many sperms are formed from one spermatogenesis process or from one spermatogonia? That is 4. Then coming to the questions related to ovum, when the ovum is released from the ovary, it is released in metaphase stage. What is the size of ovum? It is a large cell measuring about 100 micrometer. What is the approximate number of ovarian follicles present in each ovary at puberty? Approximately 40,000. And what is the approximate number of ovarian follicles that undergo ovulation during the entire reproductive period of the life? That is approximately 400. And how many ova they are formed from single ovogonium? That is 1. So from the single spermatogonium 4 sperms are formed, and from the single ovogonium 1 ovum is formed. Then is same, that is what are the stages of oogenesis? From ovogonium primary oocyte is formed, then secondary oocyte with first polar body, then ovum with the second polar body is formed. How many polar bodies are formed in one oogenesis process? That is 3. What are the polar bodies? These are the bodies having half the number of chromosome but scanty cytoplasm. So they are just for reducing the number. So polar bodies are formed. That is when primary oocyte completes its first meiotic division at the time of ovulation. So when the second meiotic division is completed, it is soon after fertilization. So this is you can say these are the important questions which can be asked from that model. Coming to this model, which is showing the development of pharyngeal clefts and pouches. So on this side the clefts are shown, on this side the pouches are shown yellow colored. So first, second, third and fourth. So this white color 1, 2, 3, 4, these are the pharyngeal arches. This blue color structure, that is, these are the clefts. Now this first arch is giving the mandibular process, then epicardial ridge is visible. So this is on this side. So what are the different structures which are arising from these pharyngeal pouches are? From this first pouch, that is primitive tympanic cavity and auditory tube is formed. So contribution of the second is also coming here. Then from second, the palatine tonsil is there. No doubt it is contributing to these structures too. That is tubotympanic recess. So tubotympanic recess is further divided into these two important structures. Then from the third inferior parathyroid gland is formed, and thymus is formed. From the fourth pouch, parathyroid, that is superior parathyroid is formed, and then you can say the some part of the thyroid is formed, which along with the ultimobranchial body, it forms the C cells of or parafollicular cells of the thyroid gland. So you can see here, so these are the different structure. Then on this side, from the first cleft, external auditory meatus is formed. So keeping in mind this model, the question can be asked, so what are those? What are the derivatives of the first pouch? What are the structure which is derived from tubotympanic recess? Because from the first pouch, dorsal part receives its contribution from the second pouch. So dorsal part and the second pouch, they form the tubotympanic recess, and that tubotympanic recess is forming auditory tube, middle ear including tympanic antrum. Then what are the derivatives of second pouch, third pouch, fourth pouch, and ultimobranchial body? So these are the important questions which can be asked from this model. So answers are given in this. Then what are the derivatives of first cleft? As I have told you, that external auditory meatus. So from which structure the pinna of the ear develop? That is swelling arising from the first and second arches adjoining first cleft. Then what is cervical sinus? It is a space between the second arch and third, fourth, sixth arch. So that is cervical sinus. Now what is pharyngeal apparatus? You know it, that pharyngeal apparatus consists of arches, clefts, pouches, and pharyngeal membrane. Next is, if a question comes, or suppose on the model, if the pharyngeal arches are asked, so there are chances they can ask you what are the derivatives of first pharyngeal arch. So answer is given over here. You should know the nerve of the first arch, which is mandibular nerve. Then what are the derivatives of the second arch? So these are the muscular component arising from it. This is the skeleton component which is arising from it. So 5S, you remember this from the second arch. So that is stapes, styloid process, stylohyoid ligament, small cornu of hyoid bone, and superior portion of the hyoid bone. So nerve is facial nerve. Then derivatives of third arch, then derivative of fourth and sixth arch. So skeleton component is laryngeal cartilages, mind it. So all the laryngeal cartilages, they are arising from fourth and sixth arch except epiglottis. So that is important. And fourth arch is the nerve is the superior laryngeal branch, and for sixth arch nerve is recurrent laryngeal nerve. Both are the branches of vagus nerve. Now which arch, pharyngeal arch, has double nerve supply? You know it, it is the first arch, mandibular nerve and chorda tympani. Which laryngeal cartilage is not developing from the fourth and sixth arch? That is epiglottis. Now coming to the next model, that is the development of the pharyngeal arches. You can see approximately at 25 weeks, 28 weeks, and 5 weeks. So these are the shown structures. They are shown in this at 25 days. What is happening? What is visible? Cranial neuropore, then caudal neuropore, then first and second pharyngeal arches are visible, then pericardial bulge, cut edge of amnion is visible, vitelline duct connecting stalk. And in this, at the 8th day, you can see the aortic placard, then lens placard is visible, then pharyngeal arches are there, then heart bulge is there, then umbilical cord. So they are visible. Then is from the aortic placard, you can see here clearly it is visible in this model. Then pharyngeal clefts are visible, then lens placard is visible, limb bud is visible, then pericardial swelling is visible in this model. So these are the labelings of the structures which are visible in this model of the development of pharyngeal arches. Now whatever be the structure is visible over here, so question can be asked on that. So lens placard, aortic placard, they are visible. So what are the structure arising from lens placard and aortic placard? From lens placard, the lens of the eye is formed. From aortic placard, the ectodermal thickening is, basically it is ectodermal thickening, invaginate to form the aortic vesicle, which will develop into the structures related to the hearing and equilibrium. So in short, it is contributing to the development of ear. Then this is the next model showing the migration of the thymus, parathyroid, and ultimobranchial body. So which structure is visible over here? So this is the ventral side of the pharynx. So side view is taken. Then this is the thyroid gland, foregut. Now on this side, if we see from the first pouch, auditory tube, tympanic cavity, and from the first cleft, primitive external acoustic meatus is visible. Then palatine tonsil is visible from the second pouch. Then superior parathyroid is arising from the fourth pouch, and inferior is arising from the third pouch. But due to migration So from this inferior thyroid along with the thymus, they migrate downward. So the name is given inferior parathyroid and superior parathyroid. Otherwise, they are developing from the inferior is arising from the third pouch, but superior is arising from the fourth pouch. But because of the migration, so the name is given superior and inferior respectively. Then ultimobranchial body is visible over here. So this is a model. Now, keeping in mind this model, what are the important structures which can be asked is that thyroid gland originates in the middle of at the level of foramen cecum, then it descends downward at the level of first tracheal ring. Third pharyngeal pouch dorsal region is forming inferior parathyroid and thymus. Fourth pharyngeal pouch dorsal is forming superior parathyroid and ventral is forming ultimobranchial body. So ultimobranchial body is forming the parafollicular cells or C cells of the thyroid gland which secrete the calcitonin. So this is model 7. Coming to the model 8 showing the development of the tongue. You can see here the lateral lingual swelling, tuberculum impar, foramen cecum is visible, copula, laryngeal orifice is there. So this development of tongue is shown at 4th week, 5th week and 6th week and 7th week. But as the development is happening, you can see the enlargement of the lateral lingual swelling is happening. Then tuberculum impar is enlarged, foramen cecum, epiglottic swelling is visible, laryngeal orifice, arytenoid cartilage is visible. In this, at 7th week, the epiglottis cartilage is also visible. So, and palatine tonsils. So this is the development of tongue. So what are the questions which can be asked from this? In this you can see, so these are the arches, first, second, third, fourth and sixth arches. So what are the basically questions which are arising from this model is, what are the pharyngeal arches contributing to the development of tongue? That is first, third and fourth. Name the various structures from pharyngeal arches contributing to the formation of tongue. So from first arch, two lingual swellings and tuberculum impar that is arising. So kindly correct this spelling, impar, I-M-P-A-R, which forms the anterior 2/3 of the tongue. From third and fourth arch, hypobranchial eminence is there. Then hypobranchial eminence, this hypobranchial eminence is forming the posterior 1/3 of the tongue and circumvallate papillae, and its dorsal part is forming the epiglottis. So these are the structure. Now which structure forms the musculature? Both intrinsic and extrinsic muscles of the tongue, that is occipital myotome. Then what are the various congenital anomalies of the tongue? So aglossia, hemiglossia, bifid tongue, tongue tie, microglossia, lingual thyroid, ankyloglossia superior. So these are the important questions which can be asked. Keeping in mind this model, you should know so how the tongue is developing and what are the related structures which are developing along with it. Now coming to this model, that is the ventral view of the palate, gum, lip and nose. You can see here that it is showing also the various types of cleft lip. Now in first, this is the normal. Normally what is visible is uvula is visible, primitive palate, incisive foramen is visible. So in this the unilateral cleft lip extending into the nose is visible. In this it is involving the jaw. Then in this the philtrum of the lip is there, and bilateral cleft lip involving the jaw is there. In this the unilateral cleft lip involving the lip joint extending into the incisive foramen. So this is how you can see, but this portion, the cleft you can say palate, is basically only involving the, you can say, the soft palate. So bifid uvula is there. So isolated cleft palate. Then if this cleft palate is extending up to the lip, so we call it as cleft lip palate combined with the unilateral anterior cleft lip, which is reaching up to the nostril. So this is the basically ventral view of the palate, gum, lip and nose. Along with it, it is showing the various types of cleft lip. Now coming to this question, what you can say, what are the different questions which can come out from that model is, so what is shown here is, here the primitive palate is shown. So they can ask you, primitive palate develops from frontonasal process, secondary palate develops from maxillary process. So what types of cleft palate? That is complete cleft palate, incomplete cleft palate. Complete may be unilateral or bilateral, incomplete may be bifid uvula, cleft of soft palate extending up to the hard palate. So these are the basically types of the cleft palate. Coming to this model, which is showing the frontal aspect of the face, how the different structures of the face they are developing. In first, you can see a 5-week embryo, then 6-week embryo, 7-week embryo, and 10-week embryo. So these changes are seen. In this, what is visible? So mandibular prominence is visible, maxillary prominence is there, nasal pit is there, then stomatodium is there. This is the eye, future eye. In between this is nasolacrimal groove, and this elevation is the frontonasal process. Now what is happening next to it? The enlargement of this is happening. Medial and lateral nasal processes or prominences, they appear. So stomatodium is there. So in next thing, what is happening, that this maxillary process is joining the, you can see here, medial nasal process. So these two structures, that is maxillary prominence, medial nasal process, and frontonasal process, they are contributing to the upper lip, and this mandibular process is contributing to the lower lip. Then comes the medial nasal process, lateral nasal process. This is nasolacrimal groove. So in this model, the fusion between maxillary and medial nasal process is shown. So always remember that maxillary process first fuses with the medial nasal process, then they fuse with the lateral nasal process. So to separate the nasal nares or anterior nares from the stomatodium. So that is the main events, you can say the sequence of events which is happening. Now this is the front of face showing some clarity, that is nasolacrimal groove is visible, philtrum is formed. So anterior nares, stomatodium, and developing eyes, they are shown. So what are the questions which are coming out from this model is, name the processes forming the upper lip. I told you, frontonasal process, medial nasal process, and maxillary process. Name the process forming the lower lip, mandibular process. Nasal septum is developing from which structure, that is frontonasal process, and what are the different types of cleft lip. So unilateral cleft lip, bilateral cleft lip, central cleft lip, or central cleft lip is also given the name hair lip. So these are the models of which I have discussed. So in first lecture, in coming lectures I will discuss the other models also. So be precise, you identify the different structures which are visible on the model, and just focus on the questions which are running around that model. So these are the few tips for your practical. Best of luck. So next models we will discuss in next lecture. Thank you very much.